Okay, let's dive right in. We see them pop up after a rainstorm, we use them to make our bread, but what do we really know about them? Today, we're taking a detailed look into the vast and honestly pretty misunderstood world of fungi. We're going deep into the hidden kingdom of mycology, the study of fungi.
When we think of fungi, this is probably the image that pops into our heads, right? These beautiful, almost magical mushrooms sprouting up from the forest floor. And they're absolutely essential. They're the great recyclers of our planet. But what you see here, this is just the tip of the iceberg of a much, much more complex story.
And this is where it gets really fascinating. The fungal kingdom has this profound dual nature, a real Dr. Jekyll and Mr. Hyde thing going on. On one hand, fungi are completely indispensable. I mean, think about it. No fungi, no bread, no beer, and no life-saving antibiotics like penicillin. But there's a darker side. They can also cause some really serious and sometimes even deadly human infections.
So, to really understand fungi, especially when they make us sick, we first have to get clear on what they are not. We tend to lump them in with other microbes, but they are fundamentally different from bacteria. So what's the actual difference?
Well, the single biggest difference boils down to this one word: eukaryotic. Fungal cells are complex, just like our own cells. They have a true nucleus that protects their DNA and all this other sophisticated internal machinery. Bacterial cells, on the other hand, are prokaryotic, way simpler, no nucleus, and this isn't just some minor biological trivia. It has huge implications for how they live and, crucially, how we fight them.
So those internal differences lead to some critical external ones. Let's look at the cell wall. Fungi use chitin. That's the same tough stuff that makes up an insect's exoskeleton. Bacteria use something called peptidoglycan. And this leads us to the most important point on this slide: antibiotics. An antibiotic like penicillin is designed specifically to blow up that bacterial cell wall, but it's completely useless against a fungal cell wall. That's why you need an antifungal for a fungal infection. A regular antibiotic just won't do a thing.
Okay, the fungal kingdom is insanely diverse. We're talking millions of species. So to make any sense of it all, scientists classify them in two main ways. You can think of this next part as your user's guide to this hidden world. The first way is pretty straightforward. It's all about their shape, or morphology. Basically, what do they look like under a microscope? You've got your unicellular yeasts, which are just single-celled little guys. Then you have molds, which are multicellular and grow in these long, thread-like structures. And then you have the really clever ones, the dimorphic fungi, which can actually switch between being a yeast and a mold. They're the shapeshifters.
And you can really see the difference here, can't you? On one side, you have those distinct, round, single yeast cells. And on the other, you see this intricate branching network of threads, we call those hyphae, that makes up a mold. This structure completely changes how they grow and interact with the world, including our bodies.
Now, the second method of classifying fungi gets a bit more intimate. For the fungus, anyway. It's all about how they reproduce. This system is all based on the kind of sexual spores they create to make the next generation.
And this gives us four major classes, or families, if you will. You've got the zygomycetes, the ascomycetes, which includes a lot of yeasts, the basidiomycetes, those are your classic mushrooms, and then there's this fascinating mystery box category called deuteromycetes, or fungi imperfecti, which is basically a catch-all for fungi where we haven't seen a sexual stage yet. So this gives us a sort of roadmap to the whole kingdom.
All right, now that we have the biology sorted, let's pivot to the medical side of all this. When a fungus successfully infects a human, we call the disease a mycosis. And understanding these is obviously pretty critical. The key to classifying these mycoses is all about location, location, location. I'm serious, it all depends on how deep the fungus manages to dig into the body. It's a spectrum, really. It can start with superficial infections, just on the outer layers of the skin, and go all the way to deep, systemic infections that can spread through your entire body.
Let's look at a quick comparison. A superficial mycosis, like Pityriasis versicolor, stays on the very top layer of the skin. It might cause some discolored patches, but it's mostly a cosmetic issue. But a subcutaneous mycosis, that's a whole different ballgame. Here, the fungus gets in through a cut or a wound, letting it invade deeper tissues. And that can lead to a much, much more serious problem.
So to make all of this really hit home, let's zoom in and do a case study on one specific subcutaneous mycosis, a pretty wild and chronic disease called rhinosporidiosis. So what in the world is it? Well,
Rhinosporidiosis is a chronic, long-term disease. It's also granulomatous, which is just a fancy way of saying your body's immune cells try to wall off the fungus, and that creates these inflammatory lesions. Its classic sign is the growth of these large, fragile polyps that kind of look like warts, and while they can show up in other places, they most often affect the nose and eyes.
And this just says it all, doesn't it? You can see the cause and the effect right next to each other. On the left, that's the direct result, a large fleshy polyp growing inside a patient's nose. And on the right, under the microscope, that's the culprit. That's the fungus itself, made visible.
So how does this even happen? Well, the path of infection usually starts with exposure to contaminated water or dust. It's often linked to people bathing in stagnant ponds. Then, the fungus gets into the body through a tiny cut or scrape. Once it's in, it starts to grow and eventually forms those trademark polyps. Finally, the diagnosis isn't made by growing the fungus in a lab, but by actually finding its structures in a tissue sample.
And this is a huge challenge for doctors and scientists. This quote gets right to the point. You can't just take a swab of Rhinosporidium seaburi and grow it in a petri dish, like you can with so many bacteria. Because it can't be cultured, the diagnosis completely depends on a pathologist spotting it under a microscope in a biopsy.
Now let's talk geography for a second, because this number is just staggering. Over 90% of all reported cases of this disease come from just a few places: India, Sri Lanka, and parts of South America. That incredibly strong link tells us there's something about the environment in those specific regions that this fungus just loves.
So, as we zoom back out from that specific case, the big picture really comes into focus. The fungal kingdom is everywhere. It's a constant, powerful force in our world, from the ordinary mold you might find on a piece of bread to a complex pathogen like the one we just looked at.
And that's really where we end up. Fungi are this incredible paradox. They heal and they harm. They build entire ecosystems and they can destroy tissue. It really leaves you with a final thought, doesn't it? In this huge, hidden world living right under our feet and all around us, what other secrets are fungi still waiting to teach us?